Hello and welcome back to another episode of Teach Me in 10. In this episode, we're joined by Donna McDade Walker at biomodal. She will highlight how their groundbreaking multi-omic solution, Duet Evoke, reveals the power of the 6-base genome by distinguishing 5-methylcytosine from 5-hydroxymethylcytosine together with all 4 canonical bases to measure multiple modes of biology from a single 5 nanogram DNA sample in a single experiment. Hi Donna, thank you so much for joining us. So I'm familiar with genetic sequencing involving the 4 canonical bases. Please could you start off by broadly explaining 5-base and 6-base sequencing?
Yeah, of course. So as you know, your 4 bases are your A, T, G, C. It's what you started with. It's what you've inherited. Whereas 5-base and 6-base, they add in these 2 methylation marks, and they help you understand what's happening now, what's on, what's off, what's changing. So it kind of gives you this full picture. So you've got your traditional sequencing, and in normal circumstances, it's reading a static code where your cells are identical from cell to cell. And then with 5-base sequencing or 6-base sequencing, you're adding in these epigenetic marks where they're dynamic elements. So they're able to tell you how they're controlling expression without actually changing the sequence. And so if we think about 5-base, there are a few methods in which to measure this, one of which is you only add in MC or methylcytosine to the original 4 bases. A much more common way of doing it is when you add in modified cytosine, which is essentially when you've added together methylcytosine, so MC, and hydroxymethylcytosine, which is HMC. So you add them both together, you get one signal, we call it modified C. Now, there is a distinct problem with that, though, because even though they are chemically very similar, they actually have very opposing functional roles. And so when you get to 6-base, that's the step change. So we have Duet Evoke, and that's able to tell you all 4 canonical bases plus MC and independently HMC, all in one workflow, taken from one sample. You get all of your 6 bases, all on the same molecule from very low input DNA with very high accuracy.
Great, thank you. So this interview is called Beyond 5 Bases, See What Your Data's Missing. How can 6-base sequencing change researchers' insights and analysis compared to 5-base methods?
So 5-base, essentially, you can think about it as being one-dimensional. You know that some modification has happened. 6-base, though, is more 2-dimensional. You're able to tell how much methylcytosine and how much hydroxymethylcytosine you actually have. And you're able to see that they, in some cases, are moving in opposite directions. So your MC is telling you about stable repression. It's about cell memory. It's generally turning genes off. And then HMC is associated with active or poised enhancers. And it's telling you about ongoing demethylation or the early change in which you're starting to demethylate. So you can reduce that kind of down to MC is your past and your present, genes are turned off. HMC is your early future, changing or turning on. So missing out HMC, you're kind of losing some of that story. And if you add together your MC and your HMC into this mod C, you're kind of neutralizing that signal. If you've got one of them's going up and one of them's going down, you add them together, you're now back to net nothing. Or you can sometimes engulf the more subtle signals. So if one's gone up a lot and one's gone down a little bit, you end up with just one signal that's in one direction. So modified cytosine just can't see that change. And by conflating everything together, you're really losing this beautiful kind of biological dynamic story, and you're reducing it into a single digit. And we find that 6-base the data is richer and it's more actionable. So not only do you get DMRs, so differentially methylated regions, where they're regions of the genome where you have differential methylation patterns between biological samples. We also are able to show differentially hydroxymethylated regions. And because of our duet construct within our assay, we're able to provide error suppression. So you don't lose your C to T mutations, which are really important. They're the most prevalent SNV within cancer. And a lot of 5-base methods, they're not able to pick them up as easily as we can. And then also we and others have shown that you can use 6-base data to provide predictive models of gene expression and chromatin accessibility with a really strong correlation. So if you were able to set up your model, theoretically you wouldn't need to do any RNA-seq or any attack-seq within the future. And modified cytosine signals just aren't good enough to be able to do that quite so well. And it's important to note that we don't just provide an assay. We've actually built this entire solution. Like, we've got this ecosystem put together to be able to ingest all of this new data. So we're able to help our collaborators go from raw reads to results. We've also built this a tertiary analysis platform. So in addition to our analysis pipelines that we've built, we also have this tertiary analysis platform that's called Modality Explore. And this is really fast and really great bit of software that can just live on your laptop. And it can run analyses within minutes that would have typically taken hours previously.
Building on that then, why is it important to distinguish between 5MC and 5HMC? And what are the consequences of missing one or the other in disease research or diagnostics?
So the oxidation of MC into HMC tells you about early biomarker activation, as I've mentioned. And we've got the deposition of MC is repressing gene expression, so you get these deactivating regions. And it's this kind of symphony of movement where looking at everything holistically increases the power to detect change. This synergy together is much better than either one alone or just looking at modified cytosine. And we've seen this when you look at them synergistically, when you look at them independently, the MC and the HMC together. It outperforms on disease classification and on mechanistic insight. So in some work that we did, we were looking at colorectal cancer cell-free DNA samples. And we wanted to have a look at the contribution of methylation when you move from healthy control into stage 1 and then on through the other stages to stage 4. And when we looked at 5-base with this modified cytosine, it was really difficult to tell the difference between healthy control and stage 1. We could tell healthy control to stage 4, but that progression in stage 1, it was invisible. And it wasn't until we looked at MC and HMC independently, so gave it that second dimension, that we were really able to see this progression from healthy control immediately into stage 1. And this is an early signal that's needed by researchers in any sort of disease detection. We've also seen one of our collaborators at the Vanderbilt University Medical School. She was looking at progenitor cells, and she saw that HMC precedes chromatin accessibility. So the deposition of HMC was telling you that the chromatin was going to start to open, which is essentially predicting the future. And then we've got MC was showing past activity. We've also shown that if you separate the MC and the HMC signals, then you can have a look at enhancer status. So you can tell the difference between an active, a primed, and a repressed enhancer, all of which you can't do with modified cytosine. Another issue with ModC is that if HMC is the predominant signal, then you would end up with the wrong biological hypothesis. Because when people look at modified C, they look at an increase in ModC, the automatic assumption is that it's coming from MC. So it's going to tell you about repressing gene expression. It's going to tell you about deactivation. But if it's actually an HMC signal that's coming through, then it's the complete opposite is actually true. And it's going to be an area of activation. and things are going to start switching on. And so, of course, there are consequences to that. You could misclassify enhancers, you might misunderstand or overlook pathway changes, and that's going to misinform, your intervention strategies. It's going to hinder disease monitoring. And from a translational perspective, you will end up detecting disease later when progression is already progressed so much further. And that's why our customers, they're so interested in SIG-space in their areas of research, and that's across oncology, neurology, aging, reproductive health, and precision medicine.
Wow, so considering the important role of these modifications, what's the impact of the SIG-space genome on biological insight and clinical utility?
So for biological insight, I would say that the SIG-space genome is able to give you genetic variants, methylcytosine and hydroxymethylcytosine. It's able to give you fragmentomic information, all from low input cell-free DNA or genomic DNA or FFPE. And it simplifies that overall workflow, giving you this integrated views that allows you to connect all the changes, because it's linking DNA bases to how genes are controlled, which is going to give you deeper insight because you're getting earlier signals and clearer mechanisms. And because we co-read genetics and epigenetics all on the same strand, we're able to provide information on things like variant-associated methylation, which is like any instance where a direct association can be made between genetic variation and changes in DNA methylation. So things like allele-specific methylation. We're also able to provide information on enhancer state. We can enable multi-omic modeling, and that's without the need of multiple kits or multiple protocols or multiple pipelines and like trying to gather everything together and make it all make sense and summing all your errors. It's all brought together into a nice, simple package. And that allows our customers to understand basic biology. It allows them to improve tissue of origin studies or cell of origin. They're able to get information from tissues as well as blood, brain fluid studies, and they're able to create these predictive models of nascent transcription. From a clinical utility perspective, we are highly optimized for liquid biopsy. We have industry-leading sensitivity and specificity, and because you only need this small amount of sample, like either from cell-free DNA or from like a sample aspirate or something, we're perfect for those kind of needle in a haystack applications, because we're making visible these really early changes that you just don't get from modified cytosine. And so we're applicable across the entire disease application stack, from early detection to subtyping to stratification through to disease monitoring, minimal residual disease tracking, and response to therapies. In neurology, we have seen we can pick up neuronal signals in CSF. So the work that's being done at the Karolinska Institute is to be able to try and take those signatures and then be able to pick them up in blood, so that in the future you would be able to monitor neurodegenerative diseases just through blood draws. And we also have many of our customers advocating at conferences and seminars, all talking about how SIG-space is enabling them in their biomarker discoveries. It's enabling them to distinguish between disease states and understand response to treatments, which is all evidence that there is real translational potential with SIG-space. So in general, I would say, if it's important to be able to see change before it's really obvious within anyone's work, whether that be gene expression or whether it's more holistically in a tumor or a cell, a neuron, or like a whole biological system, it's really important to read the whole story, and therefore it's really important to read all SIG-spaces.
Well, that sounds really promising for improving patient outcomes. Thank you so much for your time, Donna. It's been great speaking with you.
Thank you.
This episode was brought to you by Biomodal, an omics-based life sciences technology and analytics company who deliver products that bring the dynamism of our ever-changing biology into focus. To learn more about the Duet multi-omics solution EVOC, click the links in the description. See you next time.